What if you could build pretty much anything you wanted? Not with, bricks or steel, but with the very molecule of life itself. And what if you could do it on a scale so tiny you'd need a super powerful microscope just to even see it? Well, this isn't science fiction anymore. We're going to dive into how scientists are using DNA to build things on a scale we used to only dream about. Okay, check this out. It's a smiley face, right? But it's only 100 nanometers wide. To give you some perspective, a single human hair is about 80,000 nanometers thick. So this is unbelievably small. And what's it made from? Not silicon, not plastic. It's made of DNA. Yeah, the exact same stuff that makes, well, us.
So that's the billion-dollar question, isn't it? How on earth do you take the blueprint for all living things, this long, stringy molecule that holds the code for everything about you, and somehow convince it to fold itself up into a perfect, recognizable shape like this? To really get how amazing this is, we have to back up a little. This isn't just some neat new party trick. It's really the answer to a challenge laid down decades ago, a long-held dream in the world of nanotechnology. All the way back in 1959, the brilliant physicist Richard Feynman gave this now famous talk, "There's Plenty of Room at the Bottom." He basically threw down the gauntlet to the entire scientific world. He challenged them to build things from the bottom up, arranging individual atoms and molecules one by one. And this is where it gets absolutely incredible. This DNA folding technique can create features with a resolution of just 6 nanometers. Feynman once calculated you'd need dots about 8 nanometers wide to write the entire Encyclopedia Britannica on the head of a pin. So this method doesn't just meet his challenge, it completely blows it out of the water. So if you're going to build at this tiny, tiny scale, what's your material of choice? You might think it's some new, exotic element, but nope. It's a molecule that's been on this planet for billions of years, DNA. Now, we're all used to thinking of DNA as biological code, right? The instructions for life. But the key insight here is a total shift in thinking. You have to forget about the genetic information for a second and just see DNA for what it also is, a physical object, a long, programmable, and predictable piece of string. So why DNA? What makes it so perfect for this job? Well, for starters, it's a long, stringy molecule, which is great if you want to fold something. But the real magic is that it follows super simple, super predictable rules. A always pairs with T, and C always pairs with G. It's like a chemical zipper. That predictability means you can program it. You can tell it exactly how to connect and assemble itself. So if DNA is our programmable string, how do you actually write the program? Well, the method is so clever and elegant that it's named after an ancient art form, origami. The scientific name for it is scaffolded DNA origami. And here's the core idea. You start with one really long single strand of DNA. Think of that as your big piece of paper. They call it the scaffold. Then you design hundreds of short DNA strands, and these are called staples. These little staples are the secret sauce. They're like tiny, intelligent paper clips that tell the long strand exactly where to fold. And the process itself is just beautifully simple. First, a computer figures out the folding pattern and designs over 200 of these little staple strands. Then comes the magic part. You just dump everything, the long scaffold strand and all the little staples, into one pot. You heat it up so all the strands are floating around, and then you just let it cool down. That's it. As it cools, basic chemistry takes over. Each staple finds its perfectly matching spot on the scaffold, zips into place, and forces that long strand into its final pre-programmed shape. It literally builds itself. This diagram shows exactly how it works. See that long, snaking black line? That's our scaffold, our big piece of paper. And the little colored lines are the staples. If you look closely, you can see how each colored staple is designed to grab onto 2 different parts of the black scaffold. It acts like a tiny molecular clamp, pulling distant sections together and creating a crease, just like folding paper. You do that with hundreds of these programmed staples, and you can create an unbelievably complex shape. So the theory is pretty amazing. But does it actually work in the real world? Let's take a look at some of the incredible results. Here we go. This is the proof. On the left, that's the computer's blueprint for a simple square. And on the right, that's not a drawing. That's a real image from an atomic force microscope of what they actually made. Look at that. The design is a nearly perfect match to reality. That is just breathtaking precision.
Okay, a square is cool, but what about something a little more complicated? Here, the target was a five-pointed star. And again, look at that result on the right. It's an almost perfect match to the design. This just goes to show that the method isn't limited to simple squares and circles. You can create really intricate shapes with the same amazing reliability. And they didn't stop there. They created a whole gallery of different nanoshapes, triangles, rectangles, even discs with perfectly placed little holes in them. They got even more ambitious and made a tiny map of the Western Hemisphere. They even spelled out the word DNA, using DNA itself. The level of control they have is just astounding. Now, as cool as making tiny smiley faces is, this is about way more than just microscopic art. The real power here is what these shapes can do. It's about creating a whole new kind of canvas to build on. This scientist who pioneered this method, Paul Rothemund, he said his goal was to create a nano breadboard. It's a fantastic term, but what does it really mean? Well, think about an electronics breadboard, right? It's that board with all the little holes where you can plug in resistors and LEDs to build a circuit. This DNA origami is exactly that, but for molecules. Instead of carving circuits out of silicon from the top down, you're building a scaffold from the bottom up, and then you can attach other molecules to it with incredible precision. And this is where the future applications just get wild. Imagine building electronic circuits molecule by molecule, or precisely placing tiny gold particles or carbon nanotubes to create brand new types of sensors. You could even use it to build a tiny assembly line for proteins, putting them in just the right order to see how complex biological machines actually work. We're talking about a potential revolution in computing, in medicine, in everything. So what we have here is a powerful, versatile toolkit for building almost anything we can imagine at the molecular scale. It's a real answer to Feynman's challenge for more than 60 years ago. This technology gives us a level of control over the very building blocks of our world that we've never had before. Which leaves us with one really fascinating question. Now that we can build like this, what will we choose to create?